Hey, grade 10s. Welcome to your new section of work. It is called mitosis. Um, the only alternative to this is meiosis. So mitosis is quite interesting, at least. So the whole idea of mitosis, and like, I don't know if you've ever wondered, like, when you were conceived, and mommy and daddy had their special hug, you were just a single cell. And then that single cell ended up becoming what you look like today. And you kind of wonder to yourself, like, how did I grow? And it's through this process of mitosis that has allowed you to become the person you are. So I know that we just learned about cells and the differences between the two, and to a large extent, it is all about the organelles. And now I want you to take all those organelles and store them in a little box next to you. And I just want you to be aware of the cell membrane and the nucleus, because those are the only two things, and whatever's inside of the nucleus as well. So chromosomes and chromatin network. Those are the only two things I'm actually going to be using from the previous section. And then also just bring in your DNA knowledge, because we're going to need a little bit of that from when we did organic molecules. So you see there's a slide on the first slide there. It says the only time division means the same as multiplication. And it's extremely true, because what ends up happening is we double the DNA and then we divide it. And from one cell, which is divided, we end up with two identical cells. So it's the only time where division really means the same as multiplication. And if you ever tell your mathematics teacher that, there'll be a big fight. All right, so let's just quickly go over the idea of a cell cycle. The cell cycle actually comes in three parts, but we're only going to be talking about two main parts here. The first part is called G0. So that is just like normal. The cell's doing what it needs to. It functions 100%. It's doing its daily activities. And then we get interphase and the mitotic phase. Okay, so mitotic phase, fancy word, the mitosis of the cell. So we have interphase, which is a separate phase completely to mitosis itself, okay? So mitosis is that M phase over there, and the yellow little bit with the arrow, and then the blue is interphase. And you'll see interphase itself is broken up into three separate phases. So in this lesson, we're just going to do an introduction to mitosis and interphase, and then we'll end. And then the next lesson, we'll do mitosis and cytokinesis. And also please note that mitosis, or the mitotic phase M, is known as the division phase and then interphase. So those are technically the two parts. And what is mitosis itself? So it's when one cell gives rise to two cells. And this is very female orientated. So one mother cell gives rise to two genetically identical daughter cells. Okay, so one mother cell, two daughter cells, and you can see from the bottom over there, I've given you a very simplistic view of the whole idea of mitosis. So we have one parent cell. It goes through interphase, then it hits prophase, metaphase, anaphase, and eventually telo or telophase. It depends on your pronunciation as we go through. So what is mitosis itself? DNA replication occurs beforehand to ensure that each cell gets the same DNA. And that is important, because if your DNA started changing, you might change as an individual as a whole. Like you as a species, or you as an individual in the species, might change. So we need to ensure that that DNA is exactly the same. And I will teach you in matric eventually about how that DNA replication occurs, which is very important for the process of interphase. All right, so just so that you have a quick understanding, and then maybe this is a bit complex, but we're going to go through it nicely. So the daughter cell has one full set of chromosomes. So when we talk about a set, we're talking about two in this instance. So human beings have 46 chromosomes. That means you've got 23 maternal and 23 paternal. So maternal means from your mother, and paternal means from your father. Okay, so that is what mitosis is. It's the idea to create two genetically identical daughter cells. This is extremely important, and I love asking this. So the biological importance of mitosis, it's for repair, for growth, and asexual reproduction. So let's just quickly go through those briefly. So what is repair? This is when you've cut yourself, or you've been in some sort of accident, you've broken something, I don't know, something went wrong. Your body now needs to reproduce the cells that were damaged initially, and that is through this process of mitosis. So wherever the cut or the damage is, Mitosis will occur wherever there's open bit over there. And for you smarty pantsers out there, please recognize that as much as your body is going through mitosis constantly, because cells have a certain cell life, okay, so not all cells live forever, and I mentioned to you that in the previous section of work that cells have to replenish themselves every so often. You pretty much become a new person every 7 years. So repair is not necessarily just if you get damaged, it's when things get old and need to be replenished as well. Then growth, this is obviously you need to grow, and please also now realize that this idea of growth kind of is where things start to fall apart. So you do need to grow as you get older, and then eventually it kind of needs to stop, and then we're just replacing what happens. Okay, so it's either repair or replace, so you're just replacing what you originally have. But if that replacement goes pear-shaped, so something goes wrong, so let's say you have been exposed to dangerous chemicals, you drink excessively, you smoke 100 cigarettes a day, you are in the sun constantly, you're getting sunburned, you're being exposed to dangerous fumes from the local what do you call those places, factories in the area, you can kind of mess your DNA up, which would then cause a growth called cancer, and then we'll talk about that, or just rapidly keeps on reproducing, and that eventually causes a growth. Finally, we have asexual reproduction, so this mainly happens in bacteria as well as in plants, and sometimes fungus. Okay, we'll talk about that a little bit later. But asexual reproduction is when you don't use a partner to actually produce offspring. You just rapidly go through mitosis and you produce a whole bunch of clones of yourself, and then you can grow off of the original parent plant and create new versions of yourself. Okay, so where does mitosis take place? We briefly mentioned this earlier, but in all body cells, okay? Every single body cell, this occurs. And this is also known as, so AKA, also known as, it's not a famous rapper or whatever the hell AKA is, body cells are better known as somatic cells. I need you to learn that word, for matric as well, please, okay? This also, there's a type of mitosis that takes place as well in the reproductive organs, or the gonads, so that would be the testicles and the ovaries. But we find that in different tissues, and that undergoes meiosis. So that is where one parent cell produces 4 genetically unidentical daughter cells. Okay, so I'm going to say that again. Mitosis, 1 parent cell, 2 genetically identical. Meiosis, 1 parent cell, 4 genetically unidentical cells. So let's have a look at the structure of chromosomes, because the idea of a chromosome is going to become quite important eventually when we start splitting everything apart. You're going to wonder how these chromosomes came about and what is their importance, because why do we want to change what you guys recognize as chromatin, which is that like noodle soup, or that little like those squiggly lines that you drew? How does that turn into this? And this is what this diagram kind of helps you do. So over there you can see the nucleotides, which are the monomers of DNA. Then we have the DNA backbone, which is your ribose and your phosphate ions. That eventually carries on and starts coiling. Okay, so it starts coiling around a protein called histones. And those histones wrap and wrap and wrap, and I'll show you what they look like a little bit later. But it coils and coils and coils and coils, and eventually creates 1/2 of that thing. And that whole thing is called a chromosome. So that 1/2 is called a sister chromatid, and we'll go over the semantics of that a little bit later. So when 2 sister chromatids join, we create a chromosome, and those chromosomes can be found inside of the nucleus. So this is just the basic process of mitosis again. So this is just showing the maternal DNA and the paternal DNA. So in interphase, which is where this is going to end, you'll notice that there is a light blue and a dark blue version of DNA. And that is the copied version of the DNA. So you had 1 full set, so you had 46, and then you're going to double that. Okay, so that's going to give you 92, if my mathematics is correct. I think it is. All right, so you're going to have 46, and you're going to create 92 chromosomes. And then you're going to halve them again. So before interphase, you have 46. During interphase, you have 92. At the end of telophase over there, each of those cells has 46 again. All right, so we're doubling to divide later. And by doubling and dividing, it's like, what is 2 plus 2? That's 4, and then we divide it, and then we get 2 sets of 2 as well. All right. So we're going to only end up at interphase. And I want to point something out quickly, and something I didn't do in cells previously. In interphase over there, you see those blue spikes, right at the top of that cell. And then those blue spikes split, and they split again, and then they keep on, like things grow out of them. Those are called centrioles, okay? So like we have one centriole itself, then we have another centriole next to it, and two centrioles create a centrosome. Okay, I'm going to say it again. One centriole creates one centrosome, and the centrosome splits up into its two separate centrioles on either side. Okay, and that creates the poles of the cell. So like north and south pole, the Earth, we have north and south poles, or either end poles of mitosis. All right, so we have interphase over here made out of three phases. We have G1, S, and G2. G1 and G2 I'm not phased about. There's a whole lot of stuff, because remember, like if a cell is eventually going to split into two, so like we have one cell, it needs to produce two cells that are of exactly the same size. So what those G stand for is growth. So the actual cell itself grows, and when it divides, the two daughter cells are actually almost identical in size as well. That S, however, is for synthesis, and this is the most important. This is where DNA replication takes place. So in a test or an exam, if I ask you what is the importance of interphase, you are just going to write DNA replication. The DNA replicates. The DNA has doubled. There is a doubling of DNA. DNA synthesis. I don't care as long as you understand that one full set becomes two full sets. Alright, so I want you to stop the video over here and please draw this. So we have the nucleus and the nucleolus are visible. We have a cell membrane, we have a nuclear envelope, and we have chromatin. Inside over there where you see the word chromatin, I want you to draw the two sets of identical squiggles inside of that nucleus, okay? So you're going to draw a squiggle, and right next to it you're going to have to copy that exact squiggle. And that is going to indicate to you that this DNA has doubled, or maybe use two separate colors. What is a chromosome versus chromatin? Okay, so this is what I was talking about, where chromosome is made up of a DNA histone protein complex. You don't need to know that. You just need to know that it is a mixture between DNA and protein. Okay, so you can see, chromosomes have their own centromere, and we'll get to what that is a little bit later. So we have the DNA double helix over there that starts to wind, and eventually you see those little purple balls, and those purple balls are called histones. Those histones allow the DNA to actually wind around them, and when they wind, those histones bind to each other. So now you're wrapping, then the histones are wrapping around each other, and then that group of 8 histone balls, they start winding around each other. This is called supercoiling, which you can see in the middle of that diagram, and eventually we end up with a chromosome. Now, that chromosome is joined either side of those sister chromatids. So like this one over here and this one over here have to be joined in the middle by something, and that something that joins is called the centromere. All right, so at this point I'm going to backtrack a little bit. So there's a couple of words that start with C. We have centrosome, which makes up 2 centrioles. Then we have chromosomes made out of 2 sister chromatids, and the chromosomes are made up of wound up chromatin. Okay, so there it says the chromatin is a long thin DNA fiber before it is coiled into a chromosome. So let's continue this, because now I need to like solidify what I've just put in your brain. So what is a replicated chromosome? A replicated chromosome consists of 2 strands of identical DNA called chromatids or sister chromatids. Please don't get this confused with chromatids. A lot of people write that in tests and exams, and it's a good giggle, and I will take pictures and send it to my friends. And these are held together by a centromere. So what they've done over there at the top, they've made a green squiggle, and that green squiggle is the equivalent of what they have there, which is a chromosome. Now, so what on earth? Because on the left-hand side it says chromosome, and on the other side it says chromosome. Both of those are chromosomes, but whether we call them chromosome or chromatids are completely different. So when 2 chromatids are joined together, we call them a chromosome. But if I pull one of those chromatids apart, it is now also known as a chromosome. So that chromosome, let's say that's chromosome number 2, you would have got that from your mom. Then when it doubles, you're going to have 2 sets of chromosome number 2, all right? And that is held together by that pink blob called the centromere. A chromatid is when is a chromatid a chromatid? And I spoke about this earlier. A chromatid is a chromatid as long as it is held in association with a sister chromatid at the centromere. So when two chromatids, or when 2 single chromosomes are bound together, they become chromatids. And when those chromatids join by a centromere, we create a chromosome. All right, so there's a whole lot of work in here, and I really want you guys to draw and like come to terms with this. So maybe watch this once or twice if you have the opportunity. And in the next video, we are going to start with the actual division phase, or the mitosis itself. All right, have a great day, ladies. See you soon.